Hello, everyone. My name is Dina. I'm with the Barash and Boj Labs at the University of Pennsylvania. And today I'm going to be talking to you about my project, MAJIQ-CLIN, a novel tool for the identification of Mendelian disease-causing variants from RNA-seq data. So rare disease is not rare. What we mean by that is that 400 million people around the world are suffering from over 7,000 rare diseases. Approximately 80% of those rare diseases are genetic in origin, and most of those are Mendelian. And the genetic basis of more than half of these remains unknown. So the current state-of-the-art diagnostic approach to diagnosing Mendelian rare disease is whole exome sequencing. However, the diagnostic yield of this is only about 25 to 58%, depending on the study, and many variants are missed by this approach. By contrast, RNA-seq methods have been increasingly promising and used in clinical diagnostics. RNA-seq can find many types of variants that are missed by DNA-based methods, but I'm going to be talking about alternative splicing. So first of all, what is splicing? Splicing is a process by which pre-mRNA is modified with sections removed, resulting in a mature mRNA transcript, which can then go on to become protein. Alternative splicing is the same thing, but we make a decision at that splicing step with different mature mRNA transcripts coming from the same pre-mRNA. And splicing can be different in different tissues, as well as disease, and obviously can have functional consequences. So for the discussion of splicing today, I'm going to be talking about local splicing variations as a unit of splicing, which is essentially an exon and is either target or source junctions coming from it. As well as psi, percent spliced in, which is essentially a metric of junction inclusion. So why talk about alternative splicing in Mendelian disease? Alternative splicing occurs in over 90% of human genes, and 15 to 50% of pathogenic variants actually alter splicing. Splicing aberrations have also been shown to be causal in several documented Mendelian disorders, such as muscular dystrophy and Curzon syndrome, potentially many more. So our goal from this point was to develop a tool to detect alternative splicing variants from patient RNA-seq. So here I'm going to quickly go over an overview of the architecture of MAJIQ-CLIN developed by my author, Joseph Eicher. So we start off with RNA-seq from controls and from patients. We then treat each patient individually, create splice graphs, which contain these sets of LSVs individual to the controls and to the individual patients. From here, we combine the two, which allows us to quantify variants across both, as well as detect variants unique to the patient. So we detect two types of outliers that I'll be discussing. One we've called private variants, or PLSVs, which are variants that are unique to the patient. And the other is outlier variants, or OLSVs, which are variants where the junctions are the same, but the psi is different, so the usage of the junctions is different. So we made it. What do we want in a diagnostic tool for alternative splicing? We want it to handle complex and varied splicing events. We want it to run quickly and efficiently. We want it to correct for confounding factors. We want it to find the causal genes. And we want it to have good visualization and interpretation. For the purpose of time today, I'm not going to be discussing two of these, but please ask me after the talk if you're interested. So MAJIQ-CLIN handles complex and varied splicing events. To compare performance on both types of outliers that I talked about, we created synthetic data sets, including both of these types of outliers. And we compared performance across two commonly used tools, Leafcutter MD and Fraser. Due to some issues that we experienced using Fraser, some of these plots will only be showing data for Leafcutter MD. So for outlier variants, we see that MAJIQ-CLIN outperforms Leafcutter MD, which is an analysis done by Barry Slaff from our lab, especially at in different thresholds. And for private variants, we examine performance across a number of different splicing variations. Here I'm showing specifically for alternative 5' splice sites, but the others are very similar. So we can see that MAJIQ-CLIN, again, especially at more stringent thresholds, outperforms the other tools. Especially if we include this PLSV flag, which is the dotted line at the top. That is essentially a flag to show which variants are flagged by MAJIQ-CLIN as just unique to the patient and only consider those as positives, which greatly increases our performance here. The one that's different here is intron retention, and this is mostly due to the fact that Leafcutter MD does not do intron retention. So we also compared across different values of psi. So you can see here at a 5% inclusion versus a 50% inclusion of that new variant. And you can see that at low psi, we still perform decently well and much better than the competitors. While when you increase psi, all the tools understandably improve, but I think we can still show that MAJIQ-CLIN performs quite well. We also compared runtime for MAJIQ-CLIN against Leafcutter MD across a number of different conditions. This is one. Here we can show that MAJIQ-CLIN runs a lot faster than Leafcutter MD. It does use slightly more memory, but it's still within the range of what is sort of very realistic for people to be running. And again, we tested this against different conditions with similar trends. The next thing we wanted to make sure was that MAJIQ-CLIN could actually find causal variants. So here we used a data set from Cummings et al. In 2017, which is a just very well biologically validated data set. And here, please just pay attention to the green dots, which is the gene rank. So that's what order the true causal gene was returned to by the clinician. So the lower number is better. That means it's closer to the top of the list. So MAJIQ-CLIN here solves all of these cases and ranks them quite low. So that's less for the clinician to sort through. Compared to Fraser, this is Fraser 1. The rest of the analysis is Fraser 2. So we're trying to reproduce this with Fraser 2. Fraser also solves all of these cases, but ranks them slightly higher than MAJIQ-CLIN. By contrast, we found that Leafcutter MD failed to solve several of these cases. So again, MAJIQ is kind of a good choice demonstrated on this data set. So hopefully I've convinced you that MAJIQ-CLIN can detect variants reliably, different kinds of variants, and that it is does well in biological data. So then we want to see if we can do anything with undiagnosed patients that don't currently have a diagnosis from the Undiagnosed Diseases Network. And this is something I'm currently working on, but I want to show you one case study. So here, this patient demonstrated vision-related issues, cerebellar ataxia, hypoplasia, delayed speech and language development, as well as awkward gait. And I found a splicing anomaly using MAJIQ-CLIN in a gene called polar3B, which makes a RNA polymerase 3 subunit. Mutations in this gene have been associated by CLIN-VAR with polar3 related leukodystrophy, as well as a recently found dominant negative variant. And these symptoms associated with this disease fit quite well with the patient presentation. So using MAJIQ-CLIN, you can look at it a little more. So here we have a zoom in on a visualization that we have as part of MAJIQ-CLIN, developed by the wonderful San. And in the blue here, we have the de novo patient variant that is unique to this patient. So here we can see that in this variant, we're skipping exon 23 some of the time, and I'm looking into what that might mean clinically. And I think that's very exciting. So in conclusion, I hope I've shown that MAJIQ-CLIN fulfills the need for a tool for alternative splicing diagnostics. MAJIQ-CLIN performs quite well compared to other tools currently available. It's able to find new interesting variants, and it's clinically useful. And we have visualization, which I think is a very important part of it. So in conclusion, I'd like to thank my co-authors, the rest of my lab, ISMB for this amazing opportunity, as well as my funding. Barry helped with some of the analyses shown here, but also he helped work on MAJIQ-V3, which is the back end of a lot of this pipeline. And so he has already presented his poster, but it's virtually available. So I would direct you to look into that if you're interested in the technical details. Thank you.
I have one question.
Yes.
Did you try to benchmark against rare variants? So
against what, sorry?
For Fraser, we benchmark on GTEx, where there's plenty of rare variants, so that gives a nice ground truth. Have you tried that?
So the synthetic data that we made is actually based on GTEx. So for example, for the outlier data that I showed, we used different tissues, and that's where we got those different variants from to be able to detect that. And for the synthetic data, we artificially introduced splicing variants into GTEx. So the data is based on GTEx. Yeah.
It's not a genetic variant. It's a splicing.
Yes.
Okay.
Yes.